So this is RNA metabolism. Okay. So we're going to learn about transcription, which is, of course, the DNA-dependent synthesis of RNA. And anybody know what enzyme does that? DNA-dependent synthesis of RNA. Really simple.
RNA polymerase.
RNA polymerase, right. DNA makes DNA with a DNA template. RNA polymerase makes DNA, makes RNA with a DNA template. Capping, we're going to learn about capping and splicing and RNA processing. And then at the end, we actually talk about enzymes that use RNA as a template to make DNA. Anybody want to take a guess at those enzymes? I know 2 of you have used them before. Reverse transcription, retroviruses and whatnot. Another one would be, we've all used it because it's in our cells, like that. But telomerase. Telomerase uses an RNA template to add those telomeres. So, overview of RNA function. Ribonucleic acids play 3 well understood roles, right? So, we've all heard of these, right? In messenger RNAs, what do? Deliver the code to the ribosome so we can make a protein. Transfer RNAs deliver the amino acids to the ribosome to translate the message to amino acid sequence. Ribosomal RNAs is the main, scaffolding and catalytic enzymatic function of the ribosome. So catalytic protein amino acids, and those make up the amino acids for making proteins. Ribonucleic acids play several less understood functions in eukaryotic cells, and we talked about this a little, MicroRNAs, right? And they regulate the expression of genes, basically regulate the half-life of messenger RNAs. Like if there's a gene we want to stop making proteins, or a message we want to stop making proteins out of, we'll express these microRNAs, and they'll block the expression of that. Ribonucleic acids act as genomic material in viruses. Retrovirus, of course, has RNA genome. Overview of RNA metabolism. So ribonucleic acids synthesize in cells using DNA as a template, and that's transcription. Transcription is tightly regulated in order to, control the concentration of each of the proteins being produced. So some genes are on all the time. Those are called housekeeping genes, but they're what word I'm looking for. They're expressed all the time, so they're expressed constitutively, right? Constitutively expressed genes. Other genes you're going to turn on when you need them, and off when you need them, and those are heavily regulated genes. So being mainly single-stranded, RNA molecules can fold into compact structures with many functions. Some RNA molecules can act as a catalyst. We refer to those as ribozymes because it's ribonucleic acid, they're ribozymes. So often they'll use metal ions as cofactors. You don't see too many RNA molecules bound to an NAD. Well, I guess NAD has RNA in it, right? Adenine dinucleotide. So most eukaryotic ribonucleic acids are processed after synthesis. So we make our mRNAs, we've got to eliminate introns, join the exons together. We also eukaryotic genes, you've heard of this before, having the poly-A tail at the end of a message. And they think, well, that kind of regulates the half-life, how long that message is going to be out in the cell, depending on how long that poly-A tail is. We also cap the 5' end, and that's again, that's going to have functions with how it's going to interact with the ribosome ribosomal complex when you're producing those proteins. You'll see that in the next chapter. You have cat binding proteins. They kind of coordinate with the poly-A binding proteins to bind to the small subunit on the ribosome. We'll see that not tomorrow, but a week from Monday. Past Monday. So after things. Wait. Yes. Sorry. Crazy. So transcription in E. coli, nucleoside triphosphates add to the 3' end of the growing RNA strand. Sound familiar? So yeah, the chemistry is just like DNA polymerase. It's very similar chemistry. The growing chain is complementary to the template strand. Again, the synthesis is catalyzed by RNA polymerase rather than DNA polymerase. And RNA polymerase covers about 35 base pairs along the segment of DNA. So it's bound to about 35 base pairs as it slides along. Makes an RNA copy of the DNA template. And chemistry is nearly exactly the same. Rather, can you tell me one of the differences? What's the difference? Main difference. Huh? Well, uracil is a difference, but yeah, with DNA polymerase, what the substrate was DNTPs. RNA polymerase, they're just NTPs. So you've got both 3 and 5 prime. Wait. So it's the 3 prime. I don't like this. This is the 5 prime. They're not showing you the 2 prime hydroxy. But I guess the only important thing to the chemistry here is the 3 and 5 prime for the ribose. The RNA should have another hydroxy over here, right? But chemistry is the same. The 3' hydroxy attacks the phosphate, the alpha phosphate on the incoming NTP, and you release pyrophosphate as a product. Pyrophosphate then gets cleaved by pyrophosphatases quite quickly. So that's what drives the reaction forward. Is you lose the pyrophosphate, and the pyrophosphate comes apart, and that's what drives that reaction forward. Because this, you don't get it. It's not a big negative free energy in this reaction. But again, just like DNA polymerase, RNA polymerase uses magnesium as a cofactor to help make that 3' hydroxy more of a nucleophile, right? So, features of transcription. RNA polymerase is going to bind to a sequence in the DNA known as the promoter. So promoters recruit RNA polymerase, and that's going to begin transcription. Another thing that's different than DNA polymerase or DNA is we don't need a primer to start RNA transcription. No primer necessary. Growing into the new RNA temporarily base pairs with the DNA template for about 8 base pairs. DNA duplex unwinds, forming a bubble. This is called the transcription bubble. So when you unwind, you're going to need helicases, right? RNA polymerase generates positive supercoils ahead of that. And those are going to also be relieved by topoisomerases. So you separate that DNA, generate positive supercoils. There's going to be topoisomerases to relieve those as you move along. And this is the transcription bubble. It's just the bubble where you have the template and the coding strand separated. Template, no non-template. And to make the RNA copy of that template. That's where RNA polymerase would be. So, generate positive supercoils on the front, which will have to be relieved by topoisomerases if you don't have those. You'll get to the point where you won't be able to separate those strands. Stall RNA polymerase. Just some terminology. You heard me say the template plank strand serves as the template for the RNA polymerase, and then the coding strand matches the code except for the U's, right? Of what the mRNA is being produced. And this shows you some of those. So you have the DNA non-template or the coding strand, it's going to match sequence of the transcript, whereas the template strand is what's used to make the transcript. So it's basically the similar sequence. You match sequence except for the U's instead of T's for the coding strand. So both DNA strands may encode for proteins. So just because one strand codes for protein doesn't mean it's like all the way along that. It's always the coding side. It's both sides of DNA can code for proteins, and you will find them on both sides. So I guess this figure 26.3. This shows you the genome for adenovirus. It's got a fairly large genome, 36,000 base pairs. And all the arrows are where these are RNA transcripts that are coded for by the DNA double-stranded genome of adenovirus. And you can see you got transcripts going both directions. In fact, some even overlap, right?
So those are where the promoter regions start.
So these are the actual coding regions. So the promoters are going to be just before. And if you didn't know, adenovirus is one of the causative agents of the common cold. And it has a linear genome, kind of like us. It also was involved in one of the first human gene therapy trials. It was a terrible disaster. Well, so they were trying to repair an inborn error in metabolism that somebody could live with. Replacing an enzyme in the liver. They just had to have a special diet and they'd be normal. So they made an adenovirus to deliver the corrected version of that enzyme. They gave the kid a whopping dose of the adenovirus and it caused a huge inflammatory reaction and it killed him.
Just anaphylaxis.
Yeah. Killed him, and then it was a big disaster for gene therapy. Set it back probably a decade or so. So RNA polymerase is a large enzyme with no proofreading capability. So that's another key difference between RNA polymerase and DNA polymerase. No proofreading capability with RNA polymerase. So why the difference there? Huh? Why don't we have proofreading capability here where we have it in DNA polymerase?
It's not as consequential.
Right. So RNA polymerase is just going to make one, transcript at a time. If you have one base pair different, you might have one bad RNA, which might make a couple bad proteins, and eventually those will get turned over. Whereas the DNA, you need to proofread that. It has to have high fidelity because you make a copy of that. That's what's going to code for all of the proteins and all of the RNA that a cell is going to produce in its lifetime. So you want to make sure that's as proofread as possible. Not as important for RNA. So RNA polymerase lacks the 3' to 5' exonuclease, so it has a higher error rate, about 1 out of every 10,000 or 1 out of every 100,000. Base pairs, or not base pairs, bases is going to be wrong, right? And then RNA binds to promoter regions to initiate transcription. RNA polymerase binds. And this is what we mean by promoter regions. These are some promoter regions. So we didn't talk about the subunits, right? So you got 2 alpha subunits, a beta and a beta prime subunit, and then omega subunit, right? And then here, the what? In the whole of enzyme.
Did I just move the complete enzyme?
Yeah, the complete enzyme. Whole enzyme is complete enzyme. So the whole enzyme just contains all those subunits. So the 2 alpha subunits function to assemble and bind to the upstream promoter elements. And these are going to be the up elements here. So you see there's a lot of A's and T's. And then they help to assemble the holoenzyme there at the upstream. See the slide on promoters, 26.5. The beta subunit is the main catalytic subunit, so that's where you get the addition of the bases from the NTPs. Beta prime subunit is responsible for the DNA binding, and then the sigma, is that sigma? Yeah. Sigma subunit directs the enzyme to the promoter. The omega appears to protect the polymerase from denaturation. And these are examples of different promoter regions. So you got the upstream, there's a negative 35 region, that's the consensus sequence. And you have a number of nucleotides between that and the negative 10 region. When you say negative 10, it's about 10 base pairs before the coding, where you start the coding, which is the +1. And you notice in prokaryotic genes, you're nearly always at the initial coding site starts with an A.
Is the sigma on the other side?
No.
Oh, and A six.
Yeah. Did I not finish that? So, two consensus sequences, the TATA and TGAC. They're for the sigma subunit binding, and they're called the TATA sequences. They're AT rich. So what does that tell you? They're
The lower one.
lower melting point, so it's a good place for strand separation. We're going to take those strands apart. So less energy to take them apart. AT-rich upstream promoter elements between 40 and 60 binds to the alpha subunit. The AT-rich sequences promote strand separation. You just read it off there. I just read it. These sequences govern efficacy of RNA polymerase binding and therefore affect the gene expression level. So, you guys understand what that means? So these can differ by a little bit, and if they're different by a little bit, they're going to affect how well RNA polymerase is going to bind to that. So if you get something that doesn't need to be expressed so much, you might have some changes in this consistency. So transcription, initiation, and elongation have several steps. RNA polymerase binds to the promoter, creates a closed complex, and is not unwound. So it's a closed complex when the DNA is not unwound at all, still together. So I think they show that here. So RNA polymerase core binds to the DNA promoter, and this is still a closed complex. It doesn't become an open complex until you open that bubble. So you unwind the DNA a small amount. Okay. So open complex forms region from -10 to +2 unwinds. So 10 behind the start, where you're going to start the message or the RNA, new RNA molecule. The +2 unwinds. RNA polymerase moves away from the promoter. So here, transcription's initiated. We have the open complex that is formed. Transcription's initiated. Promoter clearance followed by elongation. And once that occurs, you're going to lose the sigma. RNA moves away from the promoter. Then the sigma is replaced with the NUSA. This is the sigma subunit. Oh, it's hard to tell, right? Oh, sigma 70, but that doesn't look like the sigma here. Yeah, so this is the sigma. So sigma binds to the closed complex. Once you initiate, transcription and elongation, it comes off. NUS1 comes on, and then you continue to make that continue elongation following that with the open complex. And then transcription eventually is going to be terminated, and NUS disassociates, and the RNA polymerase is then recycled and will make another RNA molecule once it binds to another promoter, forming a close complex. But we got different types of termination also. Transcription is a major target for regulation because you're using nucleotide triphosphates for each base that you add, so that's a lot of energy intensive to make a new RNA molecule. Regulation is achieved in many ways. One way is to regulate the affinity of RNA polymerase for a promoter. That has to do with the promoter sequence. There's activator proteins that will help recruit RNA polymerase to that promoter. And there will be repressor proteins that block RNA polymerase from the promoter site also. And we'll see, I think, in one of the last chapters how it's regulated. You guys learned about the lac operon before? Yeah, it's used heavily in molecular biology as a tool. I think in the last chapter we talked about the lac operon more in detail. But now we've got made RNA. Now we got to terminate it in bacteria. And there's two different ways for termination. There's Rho-independent and Rho-dependent termination. In Rho-independent termination, we have several U's near the end of the transcript. So, and you have a self-complementary region in the transcript that forms a hairpin with 15 to 20 nucleotides before the 3' end, which makes the RNA polymerase pause and disassociate. So the U's, you guys, what base is analogous to U's in DNA? T, right? So remember, A's and T's and U's, they only have 2 hydrogen bonds each. So you have an area where it's weakly held to the DNA, and you follow with a hairpin loop structure. And this, all the U's, and the hairpin causes RNA polymerase to stall. All the U's make an easy point for the RNA polymerase to just detach from the DNA transcript and you get termination. So that's Rho-independent, doesn't require any energy, anything like that. However, Rho-dependent does require ATP for termination. So this is Rho-dependent pathway in E. coli, and basically Rho is a helicase. What are helicases, you guys? Or do you remember? Helicases, they unwind or separate strands of nucleic acids, so they separate double-stranded regions. So with Rho helicase, the Rho helicase binds to a RUT site on the transcript, and then it binds to it, and it works its way up the transcript, and as it works its way up, it requires ATP because it's unfolding any secondary structure, separating the RNA. The Rho helicase migrates along the mRNA to the elongating RNA polymerase. The Rho helicase then separates the mRNA from the DNA template, and it's an ATP-dependent process to separate the two. Rho helicase then separates the mRNA from the DNA. I just said that. Right? Then that's what terminates the transcription there. So RNA polymerase will then eventually come off. So 2 kinds: Rho-independent and Rho-dependent. The independent has a string of U's and then a hairpin structure that helps it separate from the template. The Rho-dependent uses Rho, which is a helicase, so that requires energy to separate the strands. So Rho-dependent pathway is less understood. Known common CA-rich sequence, which is the RUT site, Rho utilization element. And then the Rho protein processes until the termination site is reached. Rho protein is a helicase, binds to the RUT site and helps to separate the double-stranded RNA, DNA duplex there within the RNA. And this is the figure showing you the Rho-independent pathway. And it helps it to see it. So you have RNA synthesis, And here you see all the T's on the coding strand. So we're going to make a bunch of U's for that. And then it forms this hairpin loop structure with a bunch of U's bound, which is easier to separate from the DNA template because it's a much lower energy to rip those apart. And then the hairpin structure kind of adds, it's going to cause the RNA polymerase to stop, and it's going to want to keep going to add energy to separate these two strands. And then the mRNA is released, and then the RNA polymerase is going to stop and come off. Reform on the next promoter with C. Totally different in eukaryotes for the most part. So eukaryotes contain several distinct polymerases. We have RNA polymerase 1, synthesizes pre-ribosomal RNA, for the ribosomal RNA 28S, 18S, 5.8 RNAs. So you got one RNA polymerase that just makes ribosomal RNA. You got RNA polymerase 2, which is responsible for mRNA. So RNase 2 makes mRNA. And then you have RNase 3, RNA polymerase 3 makes tRNAs and some small RNA products. And then you have plants that have RNA polymerase 4 that's responsible for the synthesis of the siRNAs, or small interfering RNAs. And then mitochondria have their own RNA polymerase also. So some features of some promoters recognized by eukaryotic RNA polymerase. You can have various regulatory sequences way upstream of your general promoters that are going to have the, again, the ta-ta box and the initiation codon. And again, you have A's and T's often at the +1 site. Or it's an A and T followed by So eukaryotic mRNA transcription involves many proteins, relies on protein-protein contacts, many highly conserved transcription factors. RNA polymerase 2 is well studied. It's a large complex of 12 subunits. Some subunits have some structural homology to the bacterial RNA polymerase. Has a carboxy terminal domain with highly conserved repeats. And these repeats are those alcoholic groups that can be phosphorylated. Here's the life cycle of an mRNA.
Synthesis of an mRNA begins in the nucleus. There, transcription factors and coactivators bind to enhancer sequences. These factors are joined by general transcription factors, SRPs, and RNA polymerase 2 at the promoter to assemble a transcription initiation complex. When transcription begins, the carboxyl terminal domain of Pol2 becomes heavily phosphorylated. The 5' end of the growing RNA is protected from 5' exon nucleases by a cap of 7-methyl-G. A cap-binding complex and different kinds of HNRNPs bind to the RNA. The spliceosome, made up of snRNAs and proteins, removes introns from the RNA. At a polyadenylation signal, a protein complex cleaves the mRNA and adds a poly-A tail on the 3' end. A poly-A binding protein and a cap-binding complex protect the mRNA and aid the RNA's export into the cytoplasm. There, an initiation factor binds the cap, while other proteins bind the mRNA and poly-A tail. The message is now prepared for translation. Initiation factors, the 40S ribosomal subunit, and the initiator tRNA scan for the nearest AUG start codon. Translation begins when the 60S subunit binds to form the ribosome and ends when the ribosome encounters a stop codon. As translation continues, the poly-A tail shortens. Below a critical length, the mRNA loses its 5' cap and is degraded.
This just shows you basically transcription at the top, at the promoters. So here you have RNA pol 2, transcription factor 2F, transcription factor 2A, and transcription binding factors. And they all form a complex at the top box, which generates kind of aligns pol 2 at the initiation site. The RNA or the DNA gets unwound. That's the initiation. You get the initiation complex. It has an open complex at this point, closed complex here. Transcription factor 2E and 2H come off. The C-terminal domain gets phosphorylated many times, and then the elongation complex occurs, and the RNA polymerase moves along the DNA template, producing the RNA. Elongation factors will come in until you find the termination site, whereas a termination complex will come in, dephosphorylate the C-terminal tail, and then you also get elongation factors come off. Also, termination factors come in and replace those, and then come off again as everything disassociates from the template. Assembly of the RNA polymerase at the promoter. Initiation by Tata binding protein. With the promoter, Tata binding protein is part of the multi-unit unit complex. Transcription factor 2D, other proteins involved are transcription factor 2B, 2FA, or 2A, 2F, 2E, and 2H. The helicase activities is within the 2H, so that helps you remember that. Helicase 2H. Unwinds the DNA at the promoter, and then kinase activity of 2H phosphorylates the polymerase at the C-terminal domain, or the carboxy-terminal domain, changing the conformation and enabling RNA polymerase to transcribe. So elongation factors bound to RNA polymerase to enhance processivity, or just like the DNA polymerase, processivity is how long it's going to be able to stay on the template and make a copy. Elongation factors bound to the RNA polymerase, they enhance processivity and coordinate post-translational modifications. So you saw from the movie, the first modification that we made is the methyl G cap. It gets capped because you make that 5' end comes out, and you make the cap right away. So the first thing you do is make a cap, and then as introns come out, the spliceosome, we're going to splice the introns out. And then at the end, there's a polyase site that's recognized by one of the elongation factors that then adds like a couple hundred hits at the end. So that's basically it. Regulation is really complex, and we'll see that in chapter 28 where we'll talk about regulation of gene expression. RNA polymerase can be selectively inhibited, so actinomycin and acridine interchelate in the DNA and prevent transcription. So things we don't want to take. Rifampicin binds to the beta subunit of bacterial RNA polymerases. So that's how we can block their growth and kill them. Alpha amantin from the mushrooms blocks polymerase 2 and polymerase 3 of predators, but doesn't block its own polymerase. So it's adapted a toxin to make things not want to eat it. Or if they do eat it, they pay dearly. Right? Processing of mRNA dozens of proteins coordinate with each other and within the proteins, and with the proteins involved in the transport of the ribosomes. Processing includes first we add the 5' cap, then we splice out introns, rejoining the exons, then we're going to add a poly-A tail, and eventually that RNA is going to be degraded once we've made proteins out of it. So this is the overall process. So as we get transcription, the cap gets added, introns are spliced out. These are the lariats. You'll see that these get spliced out and they form a lariat shape. Because you have a 5' to 2' lariat structure. So this is all bound together. And then you have your mature messenger RNA, which is all in the process after you have the poly-A also. The 5' cap is a 7-methylguanosine. And 7-methylguanosine links to the 5' end via a 5' to 5' triphosphate link. So you have the 5' of the guanosine, you have 3 phosphates, and then the 5' of the message. And that's your methyl G cap. And the guanosine is methylated on the number 7 position. Methyl groups derived from S-adenosine methionine, or SAMe. If you've heard of that, they sell that as a supplement a lot of times. A lot of neurotransmitters also require methyl groups to be added. Have a lot of this around, you won't have a deficit. So it protects the RNA from nucleases because you're kind of putting an abnormal group on the 5' end of that nucleotide, which will increase the half-life. And forms a binding site for the ribosome. And that's because you're going to have the cap binding protein, which will coordinate with the poly-A binding protein to recruit that ribosomal site to bind to. And this is the structure what we were talking about. Your 7-methyl-G, and then you have a 5, this is the 5' of the ribosome, to 3-phosphate linkage to the 5' of the first base. So, and then sometimes that first base is going to be methylated, sometimes the second base will be methylated also. And that's your basic cap. 5 to 5' triphosphate. So introns are found in most genes in vertebrates, some in yeast, and a few in bacteria. You guys remember which ones in bacteria were the only ones that typically have introns? Not regulatory, they were the tRNA genes that code for the tRNAs. So it cuts introns out. Have the mature tRNA. So exons usually less than 1,000 base pairs in lengths. Introns can range between 50 to 20,000 base pairs in length. So you can have the majority of your gene will be introns, or could be. Some genes have dozens of introns. There's 4 classes of enzymes, introns, group 1 and group 2. Introns are self-splicing. So the sequence within the intron splices itself. So it requires no additional proteins or ATP, and nuclear, mitochondrial, and chloroplast genomes differ mainly in the splicing mechanism. Okay. You have spliceosomal introns are spliced by enormous complexes called the spliceosomes, and they're the most common introns. Use these spliceosomes. Frequent in protein coding regions of eukaryotic genes. Then you have tRNA introns are spliced by protein-based enzymes. Primary transcripts cleaved by the endonuclease. Exons are joined by ATP-dependent ligases. So we'll first look at the splicing of group 1 introns. And you have the 3' hydroxy of free guanosine, GMP, GDP, etc. is used as a nucleophile, attacks the phosphodiester bond between a U and an A at the end of the intron, and releases the first portion of U ending exon. The 3' hydroxy of the U ending exon is then attacked at the 5' end of the other piece of the exon and rejoins the pieces. This shows you the chemistry of that. So here you have the guanosine comes in and attacks the U at the end of it. You have a U and an A that's conserved here, and then the guanosine is at the other end, and then you join this U and the G and the A. This is a better picture, I think. And then you have the G and the A here. The phosphate from, or this hydroxy from the U that was cut out from here will then attack the phosphate connecting the U, and you'll splice out the neutron back there. Group 2 introns use a 2' hydroxy within the intron as the nucleophile. So the nucleophile is a 2' hydroxy of an A residue within the intron. After the first cleavage, the second rightmost piece forms a lariat intermediate with a 2 to 5' phosphodiester bond. Other features are similar to the group 1. This figure shows us that. So here you have an A within the intron that attacks the G phosphate, cleaving it, and then the OH from the uridine at the beginning or the end of the exon here attacks the uridine here, and the intron itself leaves as this lariat structure. You have a 2' to 5' phosphodiester bond that forms that lariat structure, and then the RNA all spliced together.
Did the other one form a
Say again?
Group 1? Did group 1 also form a
No, group 1 doesn't form the lariat. So it's just a free guanosine that attacks it. The spliceosome introns are removed via a large complex called the spliceosome. Spliceosome is made up of SNRPs, or small nuclear RNA, small nuclear ribonuclear proteins. The SNRP RNA is called snRNA for small nuclear RNA. snRNAs are known in eukaryotes as U1, U2, U4, U5, and U6. And the GU at the 5' end and an AU at the 3' end usually mark sites for splicing. So those are conserved sites. Let's look at the animation for that.
Primary mRNA transcripts in eukaryotic nuclei usually have intron sequences found between the exons, which are the coding regions. Introns have 5' and 3' splice sites, with conserved sequences required for splicing. GU is conserved at the 5' splice site, AG is conserved at the 3' splice site, and an A within the intron serves as a branch point critical for the splicing reaction. A large protein complex called a spliceosome removes introns from primary mRNA transcripts. The spliceosome includes 5 structures called small nuclear ribonucleoproteins, abbreviated snRNPs or SNRPs. These are designated U1, U2, U4, U5, and U6. Each SNRP contains one of a class of eukaryotic RNAs, 100 to 200 nucleotides long, known as small nuclear RNAs, snRNAs. The U1 snRNA contains a sequence complementary to those near the 5' splice site of nuclear mRNA introns, and the U1 SNRP therefore binds to this region in the primary transcript. In an ATP-dependent reaction, the U2 snRNA pairs with the intron at a position encompassing the conserved A residue. Base pairing of U2 snRNA causes a bulge that displaces and helps to activate the adenylate, which will become a nucleophile during the splicing reaction. The whole structure rearranges as U4, U6, and U5 enter the site to form an inactive spliceosome. The spliceosome rearranges, expelling U1 and U4, and leaving U6 paired with the 5' splice site and with U2. The rearranged spliceosome is now active. Although ATP is required for assembly of the spliceosome, the following splicing reactions do not seem to require ATP. The splicing reactions begin at the branch point and at the 5' splice site. At the branch point, a 2' hydroxyl group attacks the phosphoryl group of the G at the 5' splice site. The resulting lariat structure has a 2' 5' phosphodiester bond. The resulting 3' hydroxyl group on the end of the exon reacts with the 5' phosphate at the remaining intron exon border. The ligated exons are released from the spliceosome, and the SNRPs dissociate from the lariat and are recycled for another round of splicing. The lariat is later degraded.
So basically, talks about, well, the movie just go through it again. We have this. Where it binds. This is just basically telling you everything that it just told us, right? Huh? What?
This slide's it.
Is it?
Yeah.
Oh. Yeah. You're right. So this is talking about adding a poly A tail. Right? So RNA polymerase 2 synthesizes RNA beyond the cleavage signal sequence. Cleavage signal is bound by an endonuclease and a polyadenylate polymerase bound to the C-terminal domain of RNA polymerase. Endonuclease cleaves RNA about 10 to 30 nucleotides downstream of the highly conserved AAUAA, which is the polyase site, as you always hear about. And then polyadenylate polymerase synthesizes, just adds a number of A's at the end.
Does the length of the A's determine how long the mRNA codes for each?
Yeah. Yeah, the more A's, the longer the half-life of that mRNA. Meaning the more protein will be produced from that mRNA. This shows you that. Right, so we have the RNA polymerase, polyadenylation factors, the AAUAA comes off, endonuclease cleaves off the rest of that. You have the AAUAA. And the polyadenylate polymerase just adds multiple A's. That's that. And then the overview of mRNA processing. This is using ova-albumin gene as an example. The gene itself is 7,700 base pairs. Primary transcript, remember initially you get that 5' cap. These exons are going to be cut out as they're being transcribed. And eventually you end up, the extra RNA at the end cleaved out, and we end up with a poly A tail and about 2,000 nucleotides, 1,872 nucleotides in that message RNA. So, total loss of about almost 6,000, not quite 6,000 base pairs. Bit interesting. So a single gene can yield different products depending on the RNA processing. So that's just your general RNA processing, what we just looked at. But some RNA, can be edited, bases removed or added. And cleavage and polyadenylation patterns can vary, yielding different mature transcripts. Immune globulin heavy chain gene is different degrees of polyadenylation can and different cleavage sites yield diverse sequences. Calcitonin and calcitonin gene-related peptide in the rat thyroid and brain, respectively, made from the same mRNA. Another protein that's made from the same mRNA, just different processing, is gelsolin versus plasmid gelsolin. And you get, with plasmid gelsolin, you just get a secretory sequence added to the N-terminus of that protein, so it's directed out of the cell, whereas cellular gelsolin, that's cut out of the transcript via transcriptional, alternative RNA processing, and that one stays within the cell, or gets sent out of the cell instead of assemble the actin cytoskeleton. This shows you an alternative splicing mechanism. In one case, you get primary transcript, and you have cleavage A1, A2, cleavage and polyadenylation at A1. You can have cleavage and polyadenylation at A2. So different cleavage points where you get the polyadenylation can be one sort of post-translation, or not post-translation modification, post-transcriptional modification. Another way to get two different transcripts from one primary transcript is altering the splicing of the introns. Alternative splicing, which leads to different mature mRNAs. You see this one? We cleaved off both the intron and this portion, and this one we just cleaved off the intron, so we end up with two different proteins. And this shows you alternative splicing in rat calcitonin. So in this case, you have a polyase site after exon 4, and in this case, it goes right through the polyase site, and you don't use the polyase site until after intron 6 or exon 6, and you end up with two different proteins in that case. If you notice, 4 gets cleaved out here. Right? This intron goes with exon 4 and this other intron polymerase.
Is it known how that's regulated, or is it just the original?
So often gets made in different tissues, right? Like in this case, say thyroid versus brain, right? So the different tissues, they think sometimes some of the microRNAs might be involved in this also, right? So, because if you notice, the SNRP complex recognizes certain sequences where the introns and exons come together. So if you have a microRNA, in this case, that blocks the SNRP binding here, it's not going to bind until it gets to here, and then you'll combine this one together. So does that make sense?
Yeah.
So there's certain microRNAs that can affect this gene splicing. And those will be expressed at different levels in different tissues. So if you have a microRNA here, it's not going to be in the thyroid per se, so you get this alternative splicing there. So processing of tRNAs and RNAs, bases are modified in post-transcriptional reactions to produce pseudouridine. If you noticed in some of the past slides, you saw this figure in like the SNRP sequences. That's a pseudouridine. Thiouridine, dihydrouridine, these are all modified bases. They're not incorporated as those modified bases. They're post-transcriptional reactions that create these modified bases. rRNAs and tRNAs are cleaved from the longer P precursors also. This shows you some of those modified bases. So you have thiouridine, inosine, 1-methylguanosine, N6-isopentyl adenosine, ribofimidine, pseudouridine, and dihydrouridine. all different bases that are modified, from bases incorporated into the T and R RNAs. So microRNAs function in gene regulation. They're short non-coding rRNAs of about 22 nucleotides long. Bind the specific regions of the mRNA to alter translation, and they also assist in cleaving mRNAs or block mRNAs from translation. 1% of the human genome might encode for microRNAs. This is a relatively new field of studying microRNA, because they didn't really discover this until, I guess, probably the mid to late 2000s. Synthesized from larger precursors processed by two endoribonucleases, Drosha and Dicer. Long precursor-free miRNAs made in the nucleus, Drosha and the DGCR8 cleave pre-miRNA to 70 to 80 nucleotide precursor, exportin, and RAN export this precursor into the cytoplasm, where Dicer cleaves the pre-miRNA to double-stranded RNA. Complements of the miRNA are removed by helicase. miRNA is loaded onto the protein complex known as RNA-induced silencing complex, or RISC. That RISC then binds the target mRNA and causes cleavage of that mRNA, which will stop, of course, translation of that mRNA. So, mRNA complements target mRNA. miRNA is the perfect complement to the target. mRNA is cleaved. If it's not the perfect, thus it's not translated, right? The mRNA is only partially complement, translation is blocked, but the mRNA is not. Should I just get rid of that? See this light.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribozymes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonucleated protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an argonaute protein. One strand of the RNA is selected and remains bound to argonaute. This is called a guide strand. The combination of the RNA and argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
So we talked about ribozymes. Those are RNA molecules that can catalytically cleave RNAs, cleave themselves or other RNAs. 3D structure is integral to the function. It's inactive if you denature. How are you going to denature a ribozyme? How would you? Heat it up?
In the cell.
Oh? Oh, well, if you have it. So, or helicase will denature in the cell, right? So it's inactive in denatured. It shows mindless kinetics, so it's just like a regular enzyme. Saturatable, has an active site, has measurable Km, and can be competitively inhibited. Nucleophilic attack of a sugar, hydroxy on the phosphate, followed by phosphodiester bond hydrolysis is the mechanism of how it works. And they're well characterized. These self-splicing group 1 introns show this examples of 26S rRNA precursor from proteazoa tetrahydramine. RNase P is another example, cleaves precursors to the tRNA. So it's a ribozyme that cleaves other RNA. Hammerhead ribozyme cleaves RNA virusoids. Circular RNAs that are replicated by plant viruses, and so named because the 2' structure, or the secondary structure, looks like the head of a hammer. So hammerhead ribozyme. And this shows you that the hammerhead ribozyme. Does it look like a head of a hammer?
No.
Not even a little bit. So cellular mRNAs are degraded at different rates, right? Well, the RNA lifetime is one means of gene regulation. How do they do this? They're varied from seconds to hours. Typical vertebrates, mRNAs last about 3 hours. 10 turnovers per cell generation. Shorter, 1.5 minutes half-lives for bacteria mRNAs. Degradation via ribonucleases. There's a hairpin structure in the mRNA that can extend the half-life. That's going to protect the half-life of that mRNA. If you have a hairpin that doesn't allow the ribonucleases to bind and cleave it. So, retroviruses can make DNA from RNA. And we use this in the lab. We want to copy our RNA, CD in it to make a cDNA library. Right? We use reverse transcriptase. It's the enzyme that does this. Virus enters the host cell. Reverse transcriptase will make a DNA copy of the RNA. Then it degrades the RNA with RNase activity that it has, and then replaces that RNA, uses DNA as a template to make a DNA copy of the RNA. So thus RNA, reverse transcriptase is both an RNA-dependent DNA polymerase, and then it also uses DNA as a template for DNA polymerase also. DNA, once it's produced, the double-stranded DNA can be incorporated into the host DNA where that virus can just sit there for years. This shows you the life cycle for retrovirus. So you have RNA, there's a tRNA. So one thing about reverse transcriptase is it requires a primer because it's a DNA polymerase. Right? And where do you think it gets the primer? Is one of the tRNAs from one of the cells that it hosts. So there's a DNA that's used as the primer. tRNA here, the tRNA works as the primer for RNA polymerase to make a heteroduplex RNA DNA duplex, and then the RNase activity removes the RNA, you make a double-stranded viral DNA. That will be incorporated via integration factors that come along with the virus integrase. It's incorporated into the host genome where it can be transcribed, and you make an RNA genome from that. And it'll pick up a tRNA packaged into the virus, so you have the RNA genome, single-stranded RNA genome, plus tRNA primer already ready to go for the next host to make a copy, a DNA copy of itself. So, retrovirus typically contains 3 genes plus a long terminal repeat. There's GAG, or group-associated antigen. It encodes a long polypeptide that is cleaved into 6 smaller proteins. Pol encodes protease that cleaves the long polypeptide. Reverse transcriptase and integrase to insert the DNA into the host genome. And N, which encodes for the viral envelope. Long terminal repeats facilitates integration of the virus genome into the host DNA. So you get transcription of the primary transcript, and then that makes polyprotein A and polyprotein B, which requires proteolytic cleavage. You get integrase, protease, and reverse transcriptase. Polyprotein B gets cleaved, and you end up with the viral envelope protein. So reverse transcriptase catalyzes 3 reactions. RNA-dependent DNA synthesis, already covered these. RNA degradation, so once you make DNA copy of the RNA, it degrades the RNA. And then DNA-dependent DNA synthesis contains zinc like DNA polymerase and uses a primer, which is that tRNA molecule it uses as the primer. It lacks 3' to 5' proofreading, so it's going to be error prone. So it's just like RNA polymerase. Which, if you look at the evolution of the AIDS virus, it's heavily prone to become resistant to protease inhibitors. Because you start giving somebody protease inhibitors, you have an error-prone copy mechanism, you're going to select for viruses that are resistant for the drug treatments. Some retroviruses cause cancer, so they'll bring with it an oncogene. So oncogenes are genes that lead to oncogenesis, or cancer. An example is Rous sarcoma virus has the SERP gene. SERP stands for sarcoma because that's how they discovered it. You can find it's overexpressed in sarcoma. It's a cancer of the bone, fat, muscle, and it encodes a non-receptor tyrosine kinase, an enzyme that affects cell divisions. Retrotransposons are the mobile genetic elements of eukaryotes. Remember the transposons, you have transposase, those are those jumping genes in the eukaryotic DNA. Code an enzyme with homology to reverse transcriptase of retroviruses, and you're able to move between positions via RNA interference. Using their enzyme to make the DNA from RNA. Unlike bacterial transposons that move directly from DNA to DNA. So eukaryotic transposons use an RNA intermediate, which is another more more evidence that shows that these probably evolved from previous retroviral infections.
So eukaryotes of retrotransposons,
Transposons,
and
yeah.
prokaryotes of ordinary transposons.
Right. So retrotransposons, however, lack the N gene, ENV gene, so they don't form viral particles. They're just able to move around the gene. They don't code anything that's going to make up a viral glue. Examples include the Ti element in yeast and the copia element in Drosophila. So two retrotransposons that they've identified. And they can be used as tools for site-specific like insertion of genes. These are examples of the Ti. And the copia elements, they contain GAG and POL, but nothing there with an ENV here. Telomeres, of course, eukaryotes don't have circular DNA chromosomes. We have linear, so at the ends, we have to take care of those, the telomeres generated at the ends. So we have tandem repeats, usually T, 1 to 4 T's, 1 to 4 G's with an AC on the opposing strands. Can be tens of thousands of base pairs long in mammals. TG strands is longer than the complement, leaves a 3' overhang of several hundred bases. Telomerases are not easily replicated using DNA polymerases. Or telomeres, not telomerases. Telomeres aren't easily replicated. Beyond an N, there is no template for the RNA primer. Chromosomes are shortened with each generation. Right? So telomerase adds telomeric sequences to solve this problem. Telomerase has RNA with a CYAX repeat to serve as the template. So telomerase, the enzyme itself, uses an RNA primer that's built into the enzyme. Not RNA primer, RNA template that's built into the enzyme so you can add base pairs on the growing DNA strand. So telomerase binds to the 3' end of the chromosome, hangs off so the RNA template extends beyond it, and then telomerase extends the 3' end using the RNA of the enzyme as the primer. And then the gap on the bottom strand is filled with DNA. So here's a picture showing you that. So DNA polymerase is only going to go so far, right? And it's going to leave this 3' overhang that you're going to want to fill in. Telomerase will bind, and it's got a complementary region there, and we can continue to add base pairs to this 3' end bit using this as a complement, and it uses this DNA as a strand, and then still have this RNA primer here, and then DNA polymerase will fill in the rest of the gap. Okay. extend the telomeres that way with the telomerase. Does it make sense? And then that's basically it for the chapter.